Good afternoon, children of class 12th. We are again continuing with chapter 14 of Biotechnology and Applications, children. And today, children, uh, we can remember how in video 12 we talked about protoplasts, about somatic hybrids, production of hybrids, hybridization through embryo rescue, anther and pollen culture we talked about. We also, children, studied about bulbosome method of barley production, transgenic barley production, and about colchine treatment of haploids to change them into diploid plants. So this was what we studied in biotechnology and its applications in the video 12. Now in the 13th video today we study about some important applications of biotechnology in agriculture, and we see one by one many of these advancements or technological advances in the applications in biotechnology. The first one, children, is production of pest resistant plants. We know pests these days have been coming and they have been troubling the farmers, and we also know, children, that how recently there was a cricket manifestation, and so many crickets they came, and there was a locust infestation recently in Rajasthan, in many parts of UP. So we see that we must develop pest resistant plants through biotechnology and its applications. Now, Bt toxin is produced by a bacterium. What is Bt toxin? It is a toxin that is produced by a bacterium called Bacillus thuringiensis. So this Bacillus thuringiensis, or the Bt toxin producing bacteria, this has got a gene. It has got a Bt toxin gene, and this gene is taken from the bacteria and it is cloned. And many Bt toxin genes they are procured from bacteria, and they are cloned from the bacteria, and they are expressed in plants to provide resistance against insects. Like insecticides are not used because insecticides, herbicides, they are very expensive to use. So they, children, are not used. But instead of that, we use resistant plants with Bt toxin technology. For example, we use plants called Bt cotton. Then we use plants called Bt corn, Bt rice, tomato, potato, and soya bean are some important plants which are modified by biotechnology. We also see that strains of Bacillus thuringiensis produce proteins to kill lepidopteran insects. Lepidopteran insects which produce tobacco budworm, armyworm. All these are killed because of the presence of the Bt toxin in tobacco plants. We see many coleopterans like beetles, etc. They are also sensitive to Bt toxins, and they also are killed. And we see many dipterans like flies and mosquitoes, etc. They also try to infest the plants, and they cause diseases to various plants. So the Bt toxins, they help to kill these insects also, these kind of infesting insects are killed. Then we see how do insect pests die with Bt toxins. How do the insect pests die through Bt toxins? To study this, we see that the insects, they die when they ingest inactive toxin. Now, the inactive Bt toxin is in the form of crystals. This inactive Bt toxin inside the plants, it is not detrimental to the plants. Neither it is detrimental to the bacteria which produces Bt toxin. And this is an alkaline pH of their gut, which turns it into active toxin. Only when the insect guts, the intestine of insects, they come in contact with the inactive Bt toxin, the alkaline pH in the gut of insects, they turn the inactive Bt toxin into active toxin. And this active toxin makes the midgut cells of the insects to swell, and their lysis causes the insect's death. Insects die in great numbers. Then we see that specific Bt toxin genes are used against specific hosts, against different kind of pests. A specific Bt toxin gene is utilized by the genetic engineers which are producing transgenic plants. Now we see, for example, protein encoded by Cry1AC and Cry2AB. These protein encoded by Cry1AC and Cry2AB, they control the cotton bollworms. The Cry1AB also controls the borers that are made in the plants. And some Bt crops are given in the table 4 on page number 556. As we see here in this table, some important Bt crops are like brinjal, where the transgenes are Cry1AA or Cry1AC. Then we see the chickpea crops, where we can generate peas that are eaten by chicken. Here we the scientists use the transgenes as Cry1AC. Likewise in corn, the scientists have used Cry1AB and Cry1AC. In cotton crops, the transgenes used are Cry1AB, Cry1AA and Cry1AC. Then in pigeon pea, the transgenes used is Cry1AC. In potato, we used Cry1AB, Cry3AA. In rice, we produce Cry1AC and Cry2AB. For tobacco crops, we produce transgenes Cry1AB and Cry1AC. Then for tomato crops, we also get Cry1AB transgenes, so that we can cause a toxin to be inserted in these plants, so that when specific pest comes against these plants or to destroy these crops, then these genes they toxify the insects and kill them. So we see this is how we use Bt toxins. Apart from Bt toxins, children, other proteins are used with insecticidal properties like lectins, alpha amylase inhibitors, and protease inhibitors. All these are used in place of Bt toxins also. And these also help to curb the specific pests against specific crop plants. Then we see the use of RNA interference, or it is known as RNA interference. This is utilized to stop certain nematodes which work against plants and which eat away the plants. They infest the plants. So in this we utilize utilize the RNAi. The RNA interference was used to prevent nematodes. Now nematodes are certain kind of annelids, or they are certain kinds of worms that are found in the soil, and these nematodes, they infest and kill so many plants. To prevent nematode infestation, nematodes are Meloidogyne incognita. So these nematodes in plants, these nematodes are killed due to RNA interference. Now in this, what happens? The RNA interference is used as method of cellular defense in all eukaryotic organisms. And the eukaryotic organisms, they utilize this method as a defense against other pests or against their enemies. So we see this has been utilized by the scientists to produce an interference in the translation of messenger RNA. And as the translation of messenger RNA is blocked by RNAi, so the infestation of nematodes is also stopped. This method involves sticking of a messenger RNA. In this, what do we do? The sticking of a messenger RNA due to a complementary double-stranded RNA molecule. There is a complementary double-stranded RNA molecule that is introduced, and this binds and prevents the translation of messenger RNA, or the messenger RNA is silenced. The source of complementary double-stranded RNA could be from an infection by viruses. So from infection of viruses, we use the complementary double-stranded RNA, and having RNA genomes that replicate via an RNA intermediate using agrobacterium vectors. What are vectors, children? Those which are utilized for carrying these genes are called vectors. Nematodes, and these vectors here are nematodes. Specific genes were introduced into the host plants so that these host plants could stand against the infestation of nematodes. By introduction of DNA, both sense and antisense RNA, the sense and the antisense RNA are produced in the host cells. Which is the host cell? The cells of the plants. These two RNAs are complementary to each other, and they form a double-stranded RNA. dsRNA is formed by the sense and antisense. And they are formed. This initiates RNAi, or ribose nucleic acid interference, and thus silences the specific messenger RNA of the nematode. Now the specific messenger RNA of the nematode is utilized for making proteins. But because this is targeted, messenger RNA is targeted, so because of the targeting of messenger RNA by RNAi, so children, we see that the two RNAs are complementary to each other and they form a double-stranded RNA. This initiates the RNAi or the RNA interference and thus silences the specific messenger RNA of the nematode. Due to silencing of the messenger RNA in nematode, the parasite is unable to survive in a transgenic host expressing specific interfering RNA. Then we see also there is control of fungal diseases like genes for chitinase and glucanase enzymes that degrade the major constituents of fungal cell walls named chitin and beta-1,3-glucans respectively have been introduced in test crops. And these enzymes, which are those enzymes? Enzymes chitinase and glucanase. These enzymes kill the pathogens and prevent the onset of disease. So we see that transgenic tobacco plants in which chitinase and glucanase genes were introduced showed high levels of resistance to fungal pathogens. So fungal pathogens cell walls were destroyed because of the presence of chitinase and glucanase genes. So these fungus could not survive further. Then we see delayed ripening in tomatoes. How is delayed ripening in tomatoes produced? We had learnt in 10th class, children, if you remember, about ethylene, which is a plant hormone. Now the plant hormone ethylene is primarily involved in fruit ripening. Ethylene is a hormone which primarily involves in fruit ripening. So what do we do with ethylene? Biosynthesis of ethylene we know is controlled by amino cyclopropane carboxylic acid or ACC. Now this acid, amino cyclopropane carboxylic acid, this controls the biosynthesis of ethylene in plants. And we see that synthesis and ACC or amino cyclopropane carboxylic acid, they oxidize enzymes. Now, US scientists Kishore and Klee, they introduced an enzyme which degrades ACC or amino cyclopropane carboxylic acid that was instrumental in the biosynthesis of ethylene. So when the US scientists, they introduced this enzyme for degrading ACC formation, so ethylene was not produced. Transgenic tomato plants fail to accumulate ethylene in them, causing delay in fruit ripening. And because of this, these tomatoes have got higher shelf value. They can be stored for a longer time till they are transported to the markets where the people can buy them. Then we see another application of biotechnology is in crops resistant to abiotic stresses. Abiotic stresses on plant crops are drought, which is dryness, salinity, too much salt in the fields, then flooding, which means too much of water in the fields, or submergence, that means many plants, they are submerged in water. Then we see that there are abiotic stresses like ultraviolet radiation, high and low temperatures. There can be extreme temperatures, very high or very low. So all these are abiotic stresses on crop plants. These can be curbed, or these can be minimized by producing stress resistant crop plants by genetic engineering. Then we see that male sterility is produced in crop plants. Goldberg, a scientist, genetically engineered male sterility in tobacco plants, and the male sterility is now priceless boon, or it is a priceless advancement to seed companies which are engaged in hybrid seed production in crops such as sunflower, tomato, brassica, etc. We see that male sterile seeds are utilized, so that there is no pollen production from nearby plants. Only these seeds or these varieties which the farmers grow, they have all the incorporated best variations in them. They have the best nutrients in them. They are pest resistant and male sterile. So that is why they were prepared by Goldberg, by genetic engineering. Then we see another biotechnically produced crop which is known as biofortified crops. Many of the crops, they have got special nutrients in them that have been increased. How they are increased? Because of the use of transgenes, which enhance nutritional quality of several crops. For example, in Indian Agricultural Research Institute, New Delhi, has developed vegetable crops which are rich in vitamins and in minerals, etc. Let us take examples of these. Maize hybrids, maize or bhutta, maize hybrids are amino acid rich because a transgene for enhancement of amino acids has been incorporated in it. And then we see that wheat variety called Atlas 66 has been used as biofortified crop because Atlas 66 is resistant or tolerant to acidic soil and to aluminium presence in the soil. And this rice, Atlas 66, is very rich in protein. That is why this is also an example of biofortified crop. Then we have golden rice with sufficient amount of vitamin A. This also, golden rice is also biofortified crop. And this not only helps the farmers to get a good price for their crops, but it also is good to feed healthy diet to the teeming millions of our country. So children, we will continue in the next video from here. Till next video, have a good day. Thank you.